Hello, HD examiners. This is Jeff Morrow with Sierra Analytics, and I'm coming to you today with another featured feature for HDExaminer. Today's featured feature is modifications and mutations, and how HDExaminer handles them. So the basics of what you need to know about modifications and mutations in HDExaminer is here in the protein view. When you have modifications or mutations that you want to compare within HDExaminer, you make a different protein state for each one. So for example, let's suppose we're doing a mutant versus wild type comparison experiment. I've already loaded my protein sequence here, so let's go ahead and edit this protein state and call it wild type, and this one will have no modifications. And then let's go to add protein state, and this one will be mutant. And down here in this window, you'll see an area called modifications and mutations. So to add a modification or mutation, you click on this plus button. And there are three different kinds of modifications and mutations you can make. There are point modifications, global modifications, and point mutations. So what are these? Well, a point modification is a single chemical formula that's attached to a single residue of your protein. So say, for example, I wanted methylation on residue number 30. I could do that. I select the residue, I select the modification I want, I would hit OK, and it gets added to this, this table right here. You may have noticed when I first brought this up that point modification can also have a generic formula. So there are a handful of common modifications here on this list, but if what you want isn't on this list, you can simply select generic formula and type in what you want for a formula, and it lets you type in both an additive formula and a subtractive formula, because some modifications add a certain chemical formula but also take away a hydrogen or something like that. So that's what this lets you do. So point modification is a single chemical formula attached to a single known residue of your protein. A global modification means that every instance of the selected amino acid has the given formula attached to it. This is less common, but there are some experiments that do that. So you can say that every G has formulation on it, for example. That probably isn't a biologically, sound example. I picked it at random, but there it is. This is how you select. This is how you tell HDExaminer that every instance of a certain amino acid has a certain formula hooked up to it. And then the final one is point mutation. Again, you select a single residue within your protein, and you simply tell it what amino acid that residue turns into. And again, when you add one, it adds it to this list. So now we have two protein states. There's the wild type, and if you select mutant, you'll see that we have red letters here instead of black ones. That's to tell you where your modifications and mutations actually are. So that's what that does there. Now, from here, whenever you import peptides, HDExaminer will do the right thing and will look for the correct isotope clusters in both the wild type and the mutant protein states. So a question I often get from HDExaminer users is, well, can I set up, say, a modification that may or may not be there within a single protein state? And the answer is no, for two reasons. Number one is that HDExaminer does not assume that your modification doesn't change the deuteration behavior of your protein. So if you modify a protein, there's a reasonable chance that the modification will actually change how it behaves when you deuterate it. And so mixing those two states together within a single protein state would potentially yield some confusing results. And so if you're doing an experiment like that where there's a modification or a mutation that may or may not be there, you do need to set up two separate protein states so that HDExaminer can fully analyze one way versus the other. Now, that's one of the reasons why we don't allow, maybe there, maybe not modifications and mutations. The second reason is actually a much more practical one, which is that if you add a mutation or a modification to a peptide, you're changing that peptide's chemical formula, and therefore you're changing its isotope cluster. And since HDExaminer uses an isotope cluster modeling algorithm to find your peptides, it needs to make sure it's searching for the right thing, and it can't do that if there's a mutation or a modification that might be there or might not be there. So when I explain to people that no, if you want, if you have a modification or a mutation that is, maybe there or maybe not, you have to set up both two separate protein states. The next question that I frequently get is, well, I don't have separate samples. I just have one sample that has the mixed versions of this protein. What do I do? Well, it turns out HDExaminer can actually handle that. If you go to edit this protein state, here in the protein state dialog box, there is this line right here, shares peptides with another protein state. Now, when you make protein states, the default behavior is that they all share peptides, because that makes it much easier within the software to compare peptides within your pool. You can look at two uptake plots together, for example, or anything like that. But if you notice, there are actually other selections available here in this dropdown. So by default, a protein state shares peptides with the first protein state in your project. But it can also share peptides and undeuteurated experiments, peptides, non-deuterated and fully deuterated controls, and peptides in all experiments. So if you are doing a protein with modifications that may or may not be there, and they're all mixed together in your sample, then what you can do is run your time course, have your experiments, and then set up your two protein states with, in this case, wild type or mutant, or modified versus unmodified, and then tell HDExaminer to share all of the experiments. And what this will do is when you go to calculate your data, it will separate those into two protein states, but where the peptides are the same, it will use the same data and do the same calculation. And when the peptides are different because one of them is modified and one of them is not, then in the unmodified protein state, it will calculate the unmodified data, and in the modified protein state, it will calculate the modified data, which has a different isotope cluster. So this right here is how you can get around that the fact that you have to set your protein states with every modification or mutation either there or not. Now, the astute viewers out there will notice that sharing experiments in this way may still not quite work, because there's a small chance that that the behavior of a peptide that isn't covered by your mutation or modification, the deuteration behavior of that other peptide that's further away from it on your protein might actually change. So if you do an experiment like this where it shares all the data, and you're noticing that some unmodified peptides have sort of a bimodal looking distribution, then you may want to rerun your time course separately for both your unmodified and modified protein states, because it's possible that your modification is actually changing the deuteration behavior of other peptides. And HDExaminer won't be able to tease those apart unless you actually have separate data to go with those two protein states. So yeah, that's everything on modifications and mutations. As usual, if you have any questions, please contact us. And this is Jeff Morrow from Sierra Analytics. Thanks for watching.